Hello, everyone, and welcome to Per Mattsson's webinar hosted by Simulations Plus. I'm Mike Lawless, Senior Principal Scientist at Simulations Plus. In today's webinar, Per will be discussing the outstanding research that he has performed to elucidate molecular properties of cell permeability for compounds beyond Lipinski's rule of 5. I also want to thank Arlene Padron of Simulations Plus for working with Per to set up this webinar. Before I turn the meeting over to Per, I'll briefly discuss Simulations Plus and some new features in ADMET Predictor 9.5, which will be released in the next few months. Simulations Plus is a publicly traded company with stock symbol SLP on the NASDAQ Composite Index. We were incorporated in 1996 and we currently have 40 employees. We are focused on developing state-of-the-art PBPK modeling and simulation software and artificial intelligence / machine learning applications. Cognigen is a Simulations Plus company that we acquired a few years ago. They were incorporated in 1992 and currently have about 40 employees. They are focused on software development, pharmacometric services, and population PK/PD data analysis. DILIsym is a Simulations Plus company that we acquired a couple of years ago. They were incorporated in 2015 and currently have about 20 employees. They are experts in quantitative systems toxicology / pharmacology modeling. Simulations Plus is your end-to-end modeling and simulation solution provider. MedChem, and here on this slide, I just list the products and their range across the drug discovery process. So MedChem Designer is a chemical structure and drawing program. It's also included in our ADMET Predictor and Gastro Plus software packages. And each of these packages can be used in the discovery phase of the drug discovery process. ADMET Predictor can be used in early discovery to weed out compounds with unfavorable predicted absorption, distribution, metabolism, and toxicity properties. Gastro Plus is the discovery preclinical and clinical, is using discovery, preclinical, and clinical phases of the drug discovery and development process. It is a PBPK modeling platform that simulates intravenous, oral cavity, ocular, inhalation, dermal, subcutaneous, and intermuscular absorption, pharmacokinetics, and pharmacodynamics in humans and animal species. DDD Plus is the industry's leading mechanistic in vitro dissolution software formulation and analytical scientist. Membrane Plus models data measured in vitro permeability and hepatocyte experiments to capture all the relevant data. PK Plus is used to create validated non-compartmental and compartmental PK models. And then DILIsym is used to predict drug-induced liver injury using quantitative systems toxicology. The next few slides, I'll discuss some of the new features in ADMET Predictor, which will be released in the next couple months. We beefed up our MedChem Designer and by adding features from our HTPK simulation module. So in this graphic, I show verapamil. One can then click on the %F button here to predict fraction absorbed, oral bioavailability, Cmax, Tmax, area under the curve at various doses. So here's a 1 milligram, 10 milligram, and 100 milligram dose. And we also predict the volume of distribution using a PBPK model. This next slide shows a CP time curve for verapamil. So we can predict the CP time curve. And here we show 100 milligram, a 10 milligram, and a 1 milligram dose. We've also added structure sensitivity analysis for some of the models in ADMET Predictor. This allows one to visualize the contributions that individual atoms and groups make to the prediction. So in this example, we are looking at Ames mutagenicity, and it's flagged this nitro group here on this end of the molecule as contributing the most to the toxicity of this. Most of the models predict that this is Ames positive. And if you replace this nitro group with a hydrogen, then all the predictions are non-toxic. And we did this by creating a PLS model from the A&E model using ECFP fingerprints. And then we generated a PLS model using the same master data set, roughly about 7,500 compounds for each one of our models. The PLS model provides a correlation coefficient for each of the ECFP keys. And when you display the compounds in this window, each atom is colored using the sum of the coefficients of all the keys that hit that atom. We've also spent a large amount of time inputting regression uncertainty into the predictions. So you'll remember we have uncertainty predictions for classification models. Now we've added it for regression models. And so most of the models now in ADMET Predictor, the regression models, will include uncertainty estimates for each prediction. So now when you predict the aqueous water solubility in ADMET Predictor, you'll also get value for the uncertainty estimate of that prediction. And in this graphic, I'm just showing ADMET Modeler and the error bars on each one of the predictions. And so if you develop models in ADMET Modeler, you'll automatically get these uncertainty predictions. We've also beefed up the metabolite generation in ADMET Predictor and MedChem Designer. So here I show Esterase, our newly added Esterase functionality. So we developed expert rules using prodrugs to help ensure that only legitimate substrates produce metabolites. So an esterase is not going to cleave, just look for all ester functional groups in the molecule. It's going to be selective on the exact type of ester group that is. And in this example, I'm showing a prodrug that's actually a carbamate that gets cleaved by an esterase between this carbonyl carbon and the oxygen here to form this alcohol. And then this CO2 automatically comes off once it's produced to produce the primary amine here. Aldehyde oxidase models, we've also created those from literature compounds in the public domain. Here we use atomic descriptors to create the models. This creates sites of metabolism and metabolites in MedChem Designer. We've also incorporated UGT metabolism models. So we've had these models in ADMET Predictor for a while. Now we're able to show the metabolite that would be generated or glucuronidated functional groups in there. We've also developed OCT2 and BSEP transporter inhibition classification models, and this is important for toxicity. We're developing a BCRP non-substrate-substrate classification model. We also participated in a collaboration with the Japanese NIHS, where they performed a number of studies on or tested environmental compounds in Ames mutagenicity. And then they allowed the modeling community to create models. And so in ADMET Predictor 9.5, these models will be available. We rebuilt the rat fraction unbound to plasma and liver microsomal intrinsic clearance models by adding new data. We also rebuilt the BBB filter model with additional data, and we've improved our 9 pipeline pilot integration using a consultant to help us there. And now it is my honor to introduce Per Mattsson of Uppsala University. He is currently an associate professor in the Department of Pharmacy and a principal investigator in the Drug Delivery Research Division. He's also program director for the master's program in pharmaceutical modeling at Uppsala University's Faculty of Pharmacy. His research is centered on the development of new computational and experimental methods to simulate, quantitate, and visualize drug distributions at the tissue, cellular, and subcellular scales, and on applying these methods to understand how the chemical features of drug molecules determine their cellular transport and subcellular targeting via membrane permeability and transporter interactions. His research interests also include how ligands outside the traditional small molecule chemical space, in particularly unusually large bioactive molecules, can be successfully developed into drugs. And before this webinar, he said that he just had a new baby boy, his third, so we want to congratulate him on that. An opportunity to ask questions will follow the presentation, so please type your questions in using the question panes on your control panel. And now, with further ado, here is Per Mattsson.
Okay, thank you very much, Michael. I'll start sharing my screen.
Yes, we see it now.
Okay, good. So thank you very much. I'm very happy for the great collaboration that we've had with Simulations Plus over the years, and for this opportunity to share some of our research with you. So as Michael said, I'm an associate professor at the Department of Pharmacy within the let's see if I can, within the Drug Delivery Research Division. So here's a summary picture from Uppsala of the entire group. I'd like to start by acknowledging some of the key collaborators in the projects that I will talk about today. So this was first and foremost a collaboration with the group of Jan Kihlberg at the Department of Chemistry at Uppsala University. A large portion of what I will talk to you about today is also a collaboration with AstraZeneca in Gothenburg and with researchers at the Broad Institute in Cambridge, Massachusetts, the team of diversity-oriented synthesis led by Jeremy Duvall. And I will also mention a study that we did together with Giulia Caron and Giuseppe Armondi at the University of Turin. So what I will discuss today deals with molecules that are outside the traditional drug-like space as defined by the Lipinski rule of 5. And I'm sure that everyone listening in is aware of the Lipinski rule, which was originally a retrospective analysis of registered drugs and successful clinical candidates, trying to find the statistical distribution of molecular properties that were compatible with clinical success for drug molecules. So this set of rules has been expanded with additional physicochemical properties over the years, and it's been reevaluated in light of new molecules. And we still find that most of the orally available drugs are found within these limits of the rule of 5. And in the original paper, it was suggested that the reason is that when you go outside these set of physical-chemical property ranges, you have increasing risks for poor permeability, poor absorption, which limits the drug likeness of these molecules. It should be said that the data set that the original rules were decided on were based on the types of drugs that were available at that time. So the types of targets would bias the physicochemical properties that you see. And over the past couple of decades, there's been sort of a shift in the types of targets that are pursued in pharma industry. From on the left-hand side here, the traditional GPCR and enzyme targets, where you typically have a well-defined binding pocket, to other classes of targets where the binding site is less well-defined, often on the outside of the protein, sometimes in regions where the surface is relatively smooth and lacks defined interaction points. So typically, you need to increase the size of the ligand to get the same affinity and the specificity towards those more problematic targets. So Jan Kihlberg's group has done a lot of analysis of these data sets of drugs that are outside the traditional rules of the rule of 5, and they've in some of their papers, they've looked at the binding sites that these types of molecules can bind to. So if we look at the molecules that strictly conform to the rule of 5, the binding sites are typically internal in the proteins or in pocket-shaped sites. Whereas as you go farther and farther outside of the traditional drug-like space, it is possible to address more difficult binding sites that are groove-shaped, tunnel-shaped, or even flat external sites. So this is one of the reasons for going into this more uncharted chemical space. But at the same time, you run into risks of decreasing the likelihood of being able to develop a successful oral drug candidate. So as I mentioned, the majority of currently registered drugs are found within the Rule of 5 guidelines, but there are exceptions to that, and there are several drug molecules that are successfully orally administered despite having a much larger molecular weight than the Lipinski rules dictate. So why is this? Well, the original paper suggested that transporters might be at least part of the explanation for these exceptions. For drug-like molecules, we have transporters primarily from two major protein families. We have the ATP binding cassette transporters, which are efflux transporters, transporting drug molecules from the inside of the cell to the outside. And we have the solute carrier superfamily, where most of those transporters mediate cellular uptake of ligands. So the question is, how do these types of proteins affect molecules that are in the beyond Rule of 5 chemical space? So over the past decades, there's been a lot of activity in the transporter field, and we looked into the literature data to see what physical chemical properties do the substrates that are transported by these classes of transporters have. And what we see is that substrates of the ABC transporters tend to have physical chemical properties that are similar to the entire range of registered drugs, whereas substrates of the solute carrier superfamily tend to be smaller and more hydrophilic than your average drug. And this becomes even more apparent if we divide this by the different subclasses of transporters within these families. So, for instance, larger and more lipophilic molecules tend to be transported by P-glycoprotein or MDR1 in the left-hand plot here. And there's only a few examples of transporter families that accept larger molecules in the solute carrier family. So predominantly you see transport via organic anion transporting polypeptides, OATPs, for the larger molecules, whereas, for instance, organic cation transporters, organic anion transporters from the SLC22 family tend to have smaller substrates on average. So it does seem a little bit like the transporter hypothesis is not relevant for helping drugs get absorbed. It's more a problem in having efflux as you go into larger molecules. But several analyses have shown that it is possible to go outside the physicochemical boundaries of the original Lipinski rules here. So the Rule of 5 is shown in the purple color in this plot. And other analyses of beyond Rule of 5 molecules have shown that cell permeability and oral bioavailability can be obtained even if you push some of these boundaries. However, these analyses that's been done on registered drugs and clinical candidates in the extended Rule of 5 or the beyond Rule of 5 chemical space show that not all of the rules included in the Rule of 5 are equally important for cellular permeability and oral absorption. It seems that it's more tolerable to increase molecular weight than, for instance, increasing the number of hydrogen bond donors in a molecule, as one example. So, as I mentioned, there are a number of molecules that are much larger than the rule of 5 limits, that are still orally administered and orally bioavailable. And looking at some example structures here, we see that a large number of these are macrocyclic, so they contain a large ring structure. There are cases where you have a large number of cases where you have molecules that are from natural origin, but there are also completely synthetic molecules in this chemical space that are orally available drugs. But it's quite clear that macrocyclic structure is more common among these successfully oral drugs in the beyond rule of 5 chemical space. And in a collaboration then with AstraZeneca and with the Broad Institute, we wanted to analyze how the molecular structure of macrocycles impact their cellular permeability. So the Broad Institute had a data set, a library of compounds that contained roughly 20,000 different macrocycles, and we used a chemoinformatic approach to select a smaller representative subset of this library to look into the structure permeability relationships here. So these are some example structures where you see the macrocyclic core, and one important feature of this library was that the synthesis method allowed synthesis of all possible permutations of the stereoisomers here. So in some cases we have up to 4 stereocenters in the molecule, so we have 16 different combinations of stereoisomers for each molecule in the data set. And we performed a range of different experiments for these molecules. So we determined their cellular permeability in the KCO2 assay. We measured solubility, log-D, pKa's for these molecules. And what we saw was that the majority of these molecules actually had a decent permeability. They were in the range where it's possible to do structural modifications and get them into a range where you would have successful oral absorption. We saw that permeability increased with increasing lipophilicity as common for most data sets. We also saw a clear separation here of that trend by the number of hydrogen bond donors. So a certain lipophilicity, the number of hydrogen bond donors will impact the permeability. So the more hydrogen bond donors, the lower the permeability, and vice versa. What we also saw for this data set was that a large proportion of the compounds had a significant efflux ratio in the KCO2 assay. So roughly 75% of the compounds that we looked at had a more than 2-fold efflux ratio. And we saw some really significant efflux ratios as well. And this was also a trend that with increasing molecular size, the efflux tended to dominate more and more. So looking at the hydrogen bonding functions in these molecules, we see here the same trend as for registered drugs and clinical candidates in this chemical space. We see that as we increase the number of hydrogen bond donors, there is a cutoff where you don't see a good cellular permeability. And we see this already as we go into 3 hydrogen bond donors in these molecules, whereas the number of hydrogen bond acceptors is much more flexible, similar to the original rule of 5 guidelines. So the data set that we generated here allowed us to develop structure property relationships And we had data both for the apical to basolateral permeability, so the total permeability across the cell model layer. But we also had data that allowed us to generate models for the passive diffusion and for the efflux component of the permeability. And we see that the same types of molecular properties or the same functional groups have different impact on the different mechanisms here. So we see, for instance, in the top left that lipophilicity descriptors are important for cellular permeability. And it seems like this fact is dominated by an impact on the passive diffusion, whereas an increase in the polar surface area decreases the overall permeability, but it does so mostly by increasing the efflux, so limiting the permeability that way. Another thing that we saw in this data set was that in several cases, smaller stereochemical differences in the molecules could have a significant impact on the permeability of the molecule. So this is one case where we have 8 different stereoisomers for these molecules, and these 8 stereoisomers divide into 2 different groups depending on the orientation of these two functional groups. So in the case where those functional groups are in a syn configuration, the permeability is much higher than when we have the anti-configuration. And this is both the case for when we measure permeability in the absence and in the presence of efflux inhibitors. So this seems like it's a combined effect of a different interaction with the efflux transporter, but also a different membrane permeability of the molecules. And looking into the passive diffusion component here, we did a computational analysis that was supported by NMR measurements of the conformation. So we did a conformational search of the molecules. And what we saw was that these syn molecules that had the higher permeability in the top row of the structures here, when we put those molecules in a polar solvent, DMSO, we see a number of different conformations where most of the polar surface is exposed to the exterior. Whereas if we put the same molecule in a non-polar solvent, those molecules form an intramolecular hydrogen bond network. So both of the hydrogen bond donor functions are involved in an intramolecular hydrogen bond. So effectively, these molecules are able to shield their hydrogen bond donor functions and most of their polarity from the surrounding environment by forming this intramolecular bonds. And these networks of hydrogen bonds are possible to form in all of the 4 syn stereoisomers, but it's not possible, due to steric constraints, it's not possible to form this network in the anti-configuration. So there we see a much larger exposed polar surface in both of the non-polar and the polar solvent. And this is likely the reason why we see the higher membrane permeability for the syn configurations. So we wanted to test this for some other molecules that also showed large stereochemical differences in permeability. So we gave the structures to a computational chemist who did not know which one of the stereoisomers had the higher permeability. And they looked at these low energy conformations of the molecules and scored them based on the exposed polarity, whether or not it was possible to form an intramolecular hydrogen bond, and ranked those different stereoisomers in the order that they thought that the different stereoisomers would have an increasing permeability. And this was a successful approach. We see that in each of these cases, it's possible to predict the ranking of the molecules based on these structural elements. So a short summary of this section of the talk is that we see in many cases substantial differences in cellular permeability based on the stereo centers in the molecules, and we can explain the stereospecific permeability by a different ability of the stereoisomers to shield polar atoms from the surrounding environment. And we also see that in many cases this shielding is driven by formation of intramolecular hydrogen bonds in the structures. We also see that in order to have both a high permeability and a high solubility, these molecules, it's necessary that those hydrogen bonds are formed dynamically. So they shouldn't be formed regardless of which environment you put the molecule in. They should form the intramolecular hydrogen bond in a non-polar environment, but open up and expose its polarity when you put it in a polar environment. And this fact has been then coined the term chameleonic properties that's been used in more recent publications on this theme. So we were interested in looking at what are the properties that allow these larger beyond rule of 5 compounds to combine the properties that you need for oral absorption, to have a good solubility at the same time as you have a good cellular permeability. And how can we provide some design rules to design orally available drugs in this chemical space? So we looked at what's available in the chemical literature, and most of the data so far has been based on model peptide systems, typically cyclic peptides. And one approach is to shield polarity by other functional groups. So here we see one case in the top here where going from a pentamer structure to a hexamer structure increases the permeability and the oral absorption and the oral bioavailability, while also at the same time increasing the polar surface area. So, and the reason for this is that the polar atoms are better shielded in the hexamer structure. And in the bottom, you see another case where polar atoms are shielded by the addition of a larger, bulkier substituent. So this is one approach of increasing permeability by shielding polarity. And if this is a transient and an environment dependent effect, then you can have the chameleonic properties that you need in the structures. So it's been proposed that as you go into beyond rule of 5 chemical space, you really need this chameleonic properties. And one line of reasoning here is a number of different property guidelines that have been proposed for cellular permeability and solubility in this chemical space. So already in the late 1990s, our group here showed a strong correlation between the exposed polar surface area of molecules and the fraction absorbed in humans of those drug molecules. And this has been later extended to larger data sets and simplified by using the 2-dimensional polar surface area instead of a 3-dimensional exposed polar surface area. At the same time, Adrian Whitty and co-workers at Boston University proposed some guidelines for the relationship between polarity and molecular weight in drugs in the beyond rule of 5 chemical space, with the reasoning that if you increase the size of the molecule, you also need to increase the polar surface area in order to maintain a suitable solubility and a suitable lipophilicity of those molecules to avoid other types of problems that you run into with two lipophilic molecules. So you have these two sets of guidelines that the polar surface area should be less than 140 square angstrom in order to maintain cell permeability, but it also needs to be large enough in relation to the molecular weight of the molecule to provide the aqueous solubility that you need. And as you go into beyond rule of 5 chemical space with a molecular weight in the 600, 700 Daltons and above, these two rules are not compatible. So in this chemical space, in this size range, you really need some sort of conformational flexibility in order to fulfill both of the rules at the same time. So the molecules need to be able to change this conformation to expose or shield polarity in order to be cell permeable and soluble at the same time. So we looked at differences in polar surface area measurements for two-dimensional and three-dimensional exposed polar surface areas for registered drugs. And we see that within the rule of 5 chemical space, the difference is not very big. On average, you get the same two-dimensional and three-dimensional polar surface area when you're below 400 Dalton in size. But as you go into larger and larger molecules, these differences increase. So the low energy conformations tend to hide some of its total polarity from the surrounding environment. So this polarity shielding and the conformation dependent exposure of polarity seems to be a common theme in molecules as you increase in size. And one extreme case of this that has been studied quite a lot is Cyclosporine A, where if you look at this from a two-dimensional, using two-dimensional descriptors, it has a very large polar surface area, 280 square angstrom, 23 hydrogen bond acceptors, but it forms a number of different intramolecular hydrogen bonds in most conformations that you see experimentally. So looking at crystal structures of cyclosporine A, the actual exposed polar surface area is considerably smaller in the range where some of those conformations actually conform to the permeability criterion of being below 140 square angstrom. So this could be part of the explanation for why cyclosporine A is a permeable molecule. And intramolecular hydrogen bonding has also been demonstrated for other model cyclic peptides and as I showed before, for these non-peptidic macrocyclic compounds. So we wanted to look at whether or not this was a general feature in larger drug molecules. So we looked at the data set of orally available molecules in the beyond rule of 5 chemical space with a molecular weight that was outside Lipinski's rule of 5. And we looked at both natural products and de novo designed molecules in this chemical space. And we see roughly the same relationship between topological polar surface area and molecular weight as in the study by Adrian Whitty, and showing that these molecules are representative of beyond rule of 5 drugs. What we see here is that the permeability that we measure in KQ2 cells in the presence of efflux inhibitors. So this is the passive membrane permeability of these molecules. We see a relatively poor correlation with the two-dimensional polar surface area, and we see even worse correlation with the molecular weight. But looking at crystal structures of these molecules and selecting the crystal structure that exposes the minimum polar surface area, we get a much improved correlation. And we see that there are a number of different structural features that come into play in these conformational differences. So in several of the cases, we see intramolecular hydrogen bonds being formed. So in this top example here, faldapravir forms a charge-reinforced intramolecular hydrogen bond in combination with a shielding from an alkyl group that further shields this intramolecular hydrogen bond from the surrounding environment. And this effectively decreases the exposed polarity from 270 to 194 square angstrom in this molecule. We see other types of mechanisms for shielding polarity. So in telithromycin, we see sort of a capping of the polarity by folding the aryl group on top of the polar groups. And this also results in a substantial decrease in the exposed polarity in these structures. So looking at the relationship that we observed for the minimum polarity exposed conformations, we would expect that these increases in exposed polarity between the different conformations would correspond to quite substantial differences in permeability. So according to our predictions here, if faldapravir would be locked in the most polar conformation, its permeability should be roughly 200-fold lower than what we actually measure in the experiments. And on average in our data set, we see that an intramolecular hydrogen bond typically leads to a reduction in around 30 square angstrom of polar surface area. Roughly the same for aryl shielding mechanisms, whereas alkyl shielding typically leads to a smaller reduction in exposed polarity. And this in turn should then correspond to an increase in permeability. So these analyses were all based on retrospective analysis of drugs that are successful and that have permeability. But some recent examples show that it's possible to use these ideas in the design of new molecules. So this is one case where Mackman and co-workers at Gilead and some collaborating companies optimized this screening hit, Cyclosporine A, into a orally bioavailable drug using some of the design principles that we find in our retrospective analysis as well. So what they did here was to, in one of the critical steps in their optimization, they introduced the possibility for a transient intramolecular hydrogen bond in the structure. And this results in considerably increased permeability and solubility at the same time. So this is one of the promising cases where these types of structural modifications that we see in our analysis can be successfully applied in drug discovery to optimize permeability in the beyond rule of 5 chemical space. So in summary, we see that it is possible to obtain cell permeability outside the chemical space of traditional drugs outside the rule of 5 chemical space. But as you move farther and farther outside, there are other constraints that are increasing. So for instance, the constraints on lipophilicity becomes much more prominent as you go outside the traditional drug-like chemical space. We see that transporters are common in this chemical space, but the transport effect is dominated by cellular efflux and not by cellular uptake. So it seems that we have a limiting factor rather than an absorptive factor based on transporters in this chemical space. We see for the macrocycles, the non-peptidic macrocycles, that permeability is dominated by the lipophilicity and the hydrogen bonding descriptors in that data set, and that there is a limit on the number of hydrogen bond donors that is similar to the classical rule of 5 guidelines, also when in this larger chemical space. We see also substantial stereospecific effects on cell permeability that we can explain by a different ability of these different stereoisomers to shield polar atoms, and that intramolecular hydrogen bonds is a dominating mechanism behind this shielding. And we also see that as you go into larger and larger molecules, it is necessary to have an environment-dependent exposure of polarity in order to maintain solubility and permeability. So again, I'd like to acknowledge the collaborators here, the group of Jan Kihlberg, the team at AstraZeneca, and particularly Björn Over, who was a postdoc and did most of the experiments for the macrocycle project. Jeremy Deval and his team, the diversity-oriented synthesis team at the Broad Institute, and Giulia Caron and Giuseppe Armondi at the University of Turin. And I leave you with another summer picture of Uppsala.
Thank you very much, Per. That was a wonderful presentation. And we've got a couple questions from the audience, if you wouldn't mind trying to address. So this first question is, when simulating ionizable compounds, and this is simulations through molecular dynamics or conformational searches, and when you model these in a non-aqueous environment, for example, CDCl3, do you keep them in the charged state? So are the basic nitrogens protonated in these non-aqueous environments?
We in the macrocycle experiments there, we actually did both. We based the simulations on the pKa of the measured pKa's in aqueous phase first, which then resulted in them carrying a positive charge. But we did calculations for the uncharged molecule as well, and we see very similar for these molecules. We see very similar results. But that is something that would definitely affect your simulations, and something that is difficult to keep track on what the actual pKa is in the non-polar environment.
Yeah. If there's nothing to donate the proton. Okay, very good. Thank you. Then could you please comment on the non-specific binding of these beyond rule of 5 compounds and the impact on permeability measurements? So do they bind in I guess the non-specific binding?
The non-specific binding, do you mean in the experiment to acid plastics or to?
Yeah, I think maybe binding to the membrane, maybe? It's a permeability measurement.
Yeah, so I mean we've done a lot of experiments on membrane binding of drugs as well, and we see that it tracks very well with the lipophilicity of the molecule. Not so much with the size of the molecule. We haven't really looked into a large data set of these beyond rule of 5 compounds for membrane binding, but what we do see is that for a more traditional set of drug molecules, we have this impact of lipophilicity, and to some extent the cross-sectional surface area of the molecule, which led us to suggest that this is an integration into the membrane. And from that, I would assume that as you go to larger sizes of molecules, they would perhaps interact with the membrane, but not integrate as well into the membrane. So I would suspect that you might have membrane binding effects, but that those interactions would be less strong than for a smaller, more a smaller structure that is more similar to a phospholipid.
Okay, yep, where it would bind in there. All right, another question is the IMHB. I'm not sure what that means. Hydrogen bonding, I guess, is the last. Exists mainly in nonpolar environments and less in aqueous environments. How could the impact of IMHB be explained at the bilayer level?
Well, what we see...
intermolecular hydrogen bonding, I'm sorry.
So what we see for these registered drugs and clinical candidates in this range is that we see a much smaller impact of the conformation on solubility predictions. We get roughly the same correlation to solubility regardless of if we use the average exposed polarity or the maximum or the minimum exposed polarity, but we see this very clear trend that permeability is much better explained by the minimally exposed polarity. Okay. And the reasoning that we have there is that in the aqueous environment, the molecules sample conformations much quicker, but there is a physical constraint as you go into the biolayer, a physical constraint from the phospholipids. So you don't have as fast conformational sampling there. That's one part of it, and at the same time you have this this energetic cost of exposing polar functions when you're inside the non-polar center of the bilayer.
Okay. Excellent. Okay, this is kind of a follow-up question on this, but was by the same person. For Keiko-2 permeability measurements, do you sometimes see a lag time? If so, was the effect of the structure on lag time analyzed? So that's a part of the experiments.
Yes. We do see a lag time in some cases. And no, we did not really analyze that. We've based the correlations here on permeability estimates from starting after the lag time. So this is the steady state transport rate of the molecules. But that is certainly interesting to see what if there's a structural impact on the lag time itself. The lag time being probably related to binding to the membrane and compartmentalizing inside the cell before you see that molecule on the other side of the cell monolayer.
Okay. And there's two questions related to lymphatic uptake. And so it looks like it's reported that cyclosporine is absorbed via lymphatic mechanisms in the literature. And it wants to know if you evaluated this mechanism and its relationship to transporters.
No, we have not. Okay. That's the short answer to that.
Yeah, some always throws a monkey wrench into the analysis. Okay, those are all good questions. Will you be able to send me the slides and then I can distribute them to anyone that asks?
Sure.
A slide deck. Okay. Yeah. So if you want to send me an email at mlawless@simulations-plus.com, then I can forward you Per's slides to you. So that looks like all we have. There's a number of comments on the wonderful talk and presentation. So I just want to thank you again, Per, and I hope that everyone has a wonderful day, and thanks again for attending. Take care, everyone.
Thank you very much.
All right. Bye-bye.